Welcome to BioClass. Today we will discuss biotechnological application in medicine. After food, just after food and agriculture, the next important here medicine. In this field, how we can prepare the different type of antibodies, hormone, protein, which it is not possible normally. And here the first, which is genetically engineered, you can say the medicine, you can say the hormone, insulin for the diabetic patient. And the disease diabetes, it is the huge number of patients is there all over the world. So that supply of insulin is very difficult. And how this insulin will be supplied. So the first method it was there, that it is from the insulin from others animals insulin it can be. It means that from the cow, bovine insulin from cow, it is extracted, and from the slaughterhouse it is taken and it is prepared. But afterwards we here it is prepared with the help of the bacteria. And here in your book it is a very brief discussion is there, that let's just know that how from the bovine insulin it how it can be done. So look at here. Here it is the bovine insulin from the cow. In bovine insulin, if we check that the chain of the insulin is here protein, which is a very short chain amino acid. The chain is not very long. Here this one is 21 amino acid chain. And here it is 30 chain B, and chain C is 33, which is found in the bovine insulin from the cow. So this insulin is extracted from the slaughtered cattle pancreas is collected, and after that the C chain is removed. The why C chain is removed? Because in human insulin it contains only A and B, these two chains. So C chain we have to remove. So A and the B chain will remain there because its similarities are there. The same number of amino acids, same type of amino acids, everything is there. Only the C chain is removed. So that the biotechnological seizure we can use, and here the C chain is removed, and this can be used for the human being. And afterwards we have some problem is created in that bovine insulin. Normally it can work in human insulin. So we have changed from this pattern, this type of the production of the insulin, to some other type. So that type also I'll discuss a little bit in your book. It is very less it is given. So we can see this type, genetically engineered insulin. Look at here. In 1978, if I make it a little bit bigger inside, in 1978, Genentech, one company, genetic engineering technology. Here I can show you that. Look at here. This is the company. This company that they made the Hemulin. They give this the trade name as given, the genetic human insulin drug. It has produced the genetically engineered bacteria for treatment of diabetes. and is the first biotech drug to be approved by the Food and Drug Administration. Just know that this one is the first biotech drug, that is human insulin. So how it can prepared? Here two different type of plasmids are taken. So here it is PUC18, University of California, UC Stanford. This is 2700 base pairs, and here A gene, A chain, that A, just now you've seen the A and the B, this two chain. So A chain gene, it is inserted in this. Okay, and this gene is collected from the human being, not cow or not any other thing. And B chain, it is produced in other plasmids. Other plasmids it is produced, another PUC18. So these are cosmid vectors. It is placed in the bacteria. So this is the E. coli, and the multiplication with the bacterium, that you know already. Multiplication with the other protein. So when other protein is produced in the bacteria, the B chain also can produce their protein, and A chain can produce their protein. So these two are, it is collected separately. See that separately these two are collected. Plasmids of E. coli, that multiplication of A chain, and here the B chain. These two plasmid, when extracts the protein of A chain and the protein of B chain, it is there. Okay, the protein, it is of that A chain and the B chain is collected. Now it is combined and fused together, the A and B chain, the cyanogen bromide, with the help of cyanogen bromide, so that the A chain and the B chain, it is joined, and it is the human insulin. Both the plasmids, it's containing E. coli, separately it is cultured in bioreactor. You know that already these are discussed, and separately it is extracted, and after that it is combined, and in this way the hemulin, it is produced. Eli Lilly company also it is marketed with the hemulin injection in 1982. So this is the first approved biotechnic medicine in the world. Okay. Next approach come here in gene therapy. Gene therapy, here in this approach we are not making any medicine, and here we are going to the genetic level and changing the gene, so that the person recovered, and not only they are recovered for the generation after generation. It means that if we change the diabetic gene into normal gene, then this diabetes disease will not be there in generation after generation. It will stop there only. So this approach, it is the gene therapy. Here it allows the correction of the genetical defect in adult form, also in child, in the embryos, and other. It is there. So here we will describe one gene therapy which is there in your syllabus. That is the first case of the gene therapy. First clinical gene therapy was given 1994 years old girl, Ashwanti De Silva from Sri Lanka. That 4 years old girl. here suffering from adenosine deaminase deficiency. That will I'm discussing this part. Look at here, gene therapy. Gene therapy here it is. It is a collection of the method that allows the correction of the genetic defect in the child or in the embryo. Theoretically, more than 3,000 genetic disease of the human we can make it cure. You can see this. So the severe combined immunodeficiency is CID, phenylketonuria, hemophilia. So many here to see the even long list, including cancer. Cancer also the genetic effect is there. So for that adult stem cell, which is formed in the bone marrow, umbilical cord, all this. So this stem cell can make new cells, and in that way that problem will be cured. How we can do that? What is the basic procedures? What kind of vector we are using? Let's know that. Here the vector means we have to transfer the correct gene. We have to remove the defective one. So for that, here we are using retrovirus. Retroviridae group, any virus from the retroviridae group. So why we are using it? Because it is having the power to enter inside the cell. That is any virus can do it easily. And what we are doing, the disease causing gene we are removing from it. Virulent gene we are removing from it, so that this virus will not create any disease. Virus can integrate into host, human chromosome. If any cure gene is added in the viral genome, then virus can be integrated into the patient. Here, when that disease causing gene it is removed, and we are viral genome we are taking out, and we are cutting the virulent part, and next we are entering here the cure gene. It indicates that it is according to the disease it will be. And this virus, we are allowed to infect that human tissue, that is stem cells, it can infect. Okay. So why we are using this dangerous virus? Because we are using the property of the virus. The virus can easily infect the cell, some specific cell that is very easily infect, and almost every cell it goes and infects. So this power of infection, power of entering inside the cell, that is only, here it is, we are using from the virus as a vector. Now next part, we can see that the fact the first clinical therapy was successful here. 4-year-old girl, Aswanti D. Silva from the Sri Lanka, suffering from ACID. Let's know that what is ACID. When any pathogen enters inside the human body, then B lymphocytes start dividing to go against pathogen. This is very simple. You know that B lymphocyte divides. It increased in number. But for this division, B lymphocyte, one enzyme is required. For the division of the B lymphocyte, one enzyme is required for the division of multiplication. The name of the enzyme is adenosine deaminase, ADA. I think everybody have got that. The ACID, the disease when this enzyme is absent. It means that when pathogen enters, then B lymphocyte cannot divide. So for the division of the B lymphocyte, the multiplication of the B lymphocyte, adenosine deaminase ADA is required. So ADA is enzyme. Enzyme means it is comes from the certain gene, ADA gene. So ADA gene is lacking in ACID's patient. So what we have to do? ADA gene we can supply that body, so the patient can recover. Okay, so here it is done in that. Okay, just see this part. What it is done in that 4 years old girl, from the red bone marrow. Red bone marrow, it is collected from that girl and it is cultured. After that, this red bone marrow, it is cultured with engineered retrovirus. Already I told that now this retrovirus we already tagged as ADA producing gene, ADA gene, so that ADA can produce that gene. ADA producing gene, it is there and it is cultured. Then when it goes inside the cell, this ADA producing gene is attached with the bone marrow cell. Then again we are injecting back to the bone marrow, replaced to the body of that patient, I mean AD Silver's body. This method is repeated different places of the body and 3-4 times. Now here what will happen, the ADA producing gene, when they are containing in the stem cell, it can produce B lymphocyte. When it will be normal, then it can very easily register disease. Otherwise this disease ACID, it is almost like AIDS disease, because this patient will die any kind of pathogenic effect, because there is no defense system inside the body. B lymphocyte cannot do any work because the divisions is not there. So ACID patient cannot survive for long days. Here I can show you some photographs that times you can see that this small girl, the Swanti De Silva, the first people to undergo gene therapy. So she created history, and here I can show that her present photograph. It means that she is survived till now, and there is no other complication and the problem. So we can say that it is successful gene therapy. All right. So that is about gene therapy. Thank you.